Every year, pharmaceutical companies push thousands of drug candidates through rigorous animal testing. They pass the safety thresholds. Yet, when these exact same compounds enter human clinical trials, the vast majority fail. The attrition rate is staggering. Over 90% of drugs that appear safe in preclinical models collapse during human trials due to unforeseen safety or efficacy issues. This bottleneck exists because humans are not simply scaled-up mice. The biological divergence between human systems and animal proxies means an animal's reaction to a chemical is a highly unreliable predictor of a human's reaction. The clinical trial for the drug TGN-1412 illustrates this perfectly. It appeared safe in non-human primates, but triggered a sudden, severe, and life-threatening systemic immune response in human subjects. This model creates a significant capital drain. Developing monoclonal antibodies routinely costs between $650 to $750 million. That capital buys a slow process. A single development cycle can drag on for 9 years, with non-human primate studies costing up to $50,000 per animal. Relying on animal models has become a massive statistical and financial liability for modern research and development pipelines. Regulators recognize this inefficiency. Both the FDA and the National Institutes of Health are currently executing a strategic pivot away from mandatory animal testing. Their new framework is built on 3 pillars: replace, reduce, and refine. The priority sits on the first pillar, a transition from traditional animal models directly to human-relevant alternatives. The ultimate goal is to achieve safer, faster, and more ethical drug development. These human-relevant alternatives fall under a single umbrella term: New Approach Methodologies, or NAMs. The adoption of NAMs allows toxicology to move away from interspecies guesswork toward a foundation of engineered human biology. The operational goal of any NAM is precise: to evaluate systemic toxicity and drug efficacy using directly human-relevant data architectures. This begins with the in vitro layer. These tools range from standard 2D cell cultures up to complex 3D patient-derived organoids that replicate specific genetic profiles. The current pinnacle of in vitro technology is organs on a chip, known technically as microphysiological systems. These microsystems replicate exact human tissue microenvironments. As fluid flows through the chip's channels over a layer of living human cells, scientists can measure the precise biochemical response to a compound. Below the cellular level is the in-chemical layer, where protein assays are deployed to test for chemical irritancy, completely eliminating the need for living test subjects. By utilizing these physical tools, toxicologists isolate and observe human biological mechanisms that are physically impossible to accurately measure in an animal proxy. Physical NAMs generate data, but their predictive power requires scale. This scale is driven by the TOX-21 Consortium, a primary federal collaboration. TOX-21 relies on quantitative high-throughput screening. This is an industrial-scale operation. Automated robotic systems rapidly screen a library of 10,000 environmental chemicals and approved drugs against human cell lines. This continuous screening catalogs specific toxicity mechanisms, mapping exactly how thousands of compounds disrupt or interact with complex human biological pathways. All of this physical data feeds the final layer of NAMs, in silico, or computational predictive toxicology. Artificial intelligence and machine learning ingest the massive TOX-21 datasets. This allows computers to simulate human pharmacokinetics and generate a distinct probability curve for a new drug, all without conducting a single physical test. The strategic advantage here is timing. Research teams can look at these simulations and make highly accurate go or no-go decisions extraordinarily early in the development pipeline. The combination of high-throughput screening and in silico modeling means developers can fail a toxic compound in a computer simulation in seconds, rather than observing it fail in a primate over several months. Despite the science, a major procedural roadblock remains in the industry. Many developers operate under the entrenched fear that the FDA will automatically reject any submission that lacks animal data. That assumption was shattered by the passage of the FDA Modernization Act 2.0. The act explicitly changed the law to allow developers to use new approach methodologies in investigational new drug applications. For biosimilars, the impact is even more direct. The legislation removes the animal testing requirement for biologics license applications. The federal government is also actively financing this transition. The NIH recently announced over $150 million in funding through the Complement REI program to develop and standardize these human-based methods. Sponsors can now navigate the transition to NAMs by mastering specific validation pathways, as the legal and regulatory hurdles have been removed. To help drug developers integrate NAMs, the FDA established formal validation frameworks. The first is the iStand program, designed for early engagement on novel tools. To use iStand, the sponsor must meet a strict requirement. They must define a highly specific context of use for their tool. For permanent qualification, sponsors use the Drug Development Tool, or DDT, qualification program. DDT qualification requires rigorous evidence, demonstrable human relevance, reproducibility across lots, and strict predictive accuracy. The operational advantage is clear. Once a tool is qualified, it can be used in multiple IND and NDA submissions forever, without the sponsor needing to resubmit validation data. It is worth noting that if these NAM platforms are ever used to guide individual patient treatment decisions, they must also meet CLI validation standards as laboratory-developed tests. By utilizing Q submissions for early engagement and mastering these qualification pathways, R&D teams can transform new approach methodologies into a standardized, highly profitable pipeline advantage.